All right, so I stopped somewhere talking about polarities in amino acids and how you make the hemoglobin is going to have the different polar amino acids that are going to form the three-dimensional structure. So remember, the secondary structure is usually going to involve either an alpha helix, which that's mentioned in your book, right, which kind of looks like a curl, like if you're looking at Shirley Temple's curls, a curl. And the reason that that is going to have that shape is because of all this hydrogen bonding, okay, and the polar amino acids that are going to make bonds, hydrogen bonds between other polar amino acids. Or it could be a beta-pleated sheet, which is going to look kind of like a bobby pin. So it's like a 180-degree turn, right? So they are going to start the three-dimensional structure. That's called the secondary structure of proteins. The tertiary structure of proteins is going to be, remember, proteins are very long. So the sequence of amino acids could be even hundreds of amino acids long. Like insulin, for example, is over 400 amino acids long. So it's going to have parts of it is going to be alpha helix. Part of it will be beta-pleated sheet. And then the other parts of it will not have any particular structure. That means it doesn't necessarily have any hydrogen bonding. And there might be just a part that are all non-polar amino acids, right? So the tertiary structure is going to be like alpha helix, beta-pleated sheet, nothing, beta-pleated sheet, alpha helix, and so on. And so that then is, think about a protein sometimes are called globular because they look like a rolled up piece of paper. That's their three-dimensional structure. Sometimes, like hemoglobin, you're going to have two alphas and two betas. What does that mean? Wait. What does that mean? That means it has four parts that are going to make one functional protein. It's going to have two that come from one sequence of a DNA, and then another one that comes from another sequence. And you need two of each in order to make a four rolled up balls of paper in order for it to work. All right, so in the case of hemoglobin, that's what they call them. They call them alpha hemoglobin, which is usually found in people with sickle cell. And then the beta strand, the beta part, is going to be the one that's defective in people who have sickle cell anemia. So that is the one that can't keep the structure, the three-dimensional structure proper. So that then it can't carry oxygen if it does that. All right, so you have primary, secondary, tertiary, and quaternary structure of proteins. Again, some of them don't have a quaternary structure because it's only one strand. But in the case of hemoglobin, it has four strands that all have to come together. And if they don't come together, hemoglobin is not functional. Okay, so look that up in your book. Okay, so then other things that are important. Okay, so clearly your amino acids are going to be made up of carbon, hydrogen, oxygen, nitrogen, right? And your amino acids look like this. So I'm going to kind of clear this out here on the side, okay? So this is what your amino acid looks like. And it's really important that you know this, okay? It has a carbon in the middle, right? Because carbon can make up to 4 bonds, right? Because it has 4 electrons on the outer shell. Then it has a carboxyl group. So that COOH is called a carboxyl group. And it has an amino group. Okay, so this is like ammonia. Ammonia is NH3. This is an NH2. It's called an amino group. Okay, so it's 1 nitrogen, 2 hydrogens. Now, there's a hydrogen here. And then there's an R group here. And then this is the part of your amino acid that's going to change. All of the amino acids are going to have this the same. And then this one's going to be different. Okay, so that one's always going to be the same. This is always going to be the same. This is going to be what changes. These are all covalent bonds because covalent bonds are denoted by straight lines. This is a double bond. So that means they're sharing 2 pairs of electrons. Whenever you see a single bond, they're sharing 1 pair of electrons. All right, remember there's a nitrogen and there's an oxygen. And so because there's a nitrogen on one side, oxygen on the other, what ends up happening is that these end up being non-polar. The polarity of amino acid comes from the R group. So if you look in your book, it's going to have different R groups. So if you look online, it's going to have different R groups. So some of them are going to be very simple. They'll have carbon, hydrogens in this R group. Some of them are quite complex. They have ring structures and other things that are going to be much more complex. Now, the way that bonding occurs in your amino acids is that, remember, your ribosomes are the ones that do translation. So the ribosomes, they bring this amino acid close enough to the other amino acid right next to it. So another amino acid right next to it, which would have its carboxyl group on this side, like this, yes? And it'll be a C and then an NH, the amino group on that side, right? So we're just looking at one end. So because they're next to each other, these here, okay, so I don't know if you see that, it's an H, and there's an H, and there's an O. Two H's and an O. Two H's and an O form a water molecule. Okay? Water molecule leaves. That's called dehydration synthesis. This carbon, this nitrogen, they make a bond that makes something we call a peptide bond. Peptide bonds are the bonds between amino acids. It's a special type of the carbon-nitrogen bond that they make. And a water molecule is going to be released in that. So that's called dehydration synthesis, is when you put them together. When you break them apart, you call it hydrolysis. Lysis means cut, and hydro means water. So you're taking water and you're breaking it apart. So in digestion, it's a bunch of hydrolysis reactions. When you're making muscle, for example, it's a bunch of dehydration synthesis reactions, right? Because you're making the contractile proteins of muscle. Contractile proteins are made up of a bunch of amino acids, right? So there's 20 different amino acids. So you have amino acids that are, again, polar and non-polar. You have amino acids that you need. They're called essential amino acids that you have to consume from food. And then there's some that are not, that you can actually recycle in your body. And then they're, some of them are converted from other amino acids. So those amino acids are all going to be necessary. They all have to be present in order for you to be able to make a particular protein. All right. So that is proteins, right? So then let's talk about carbohydrates. Okay. So carbohydrates are sort of a different beast. So I cleared up the screen so you could see a little bit better. So this is glucose, okay? So glucose is a very important molecule, as you could probably begin to even just are starting to understand right now, right? So glucose is the source of all of our, pretty much the main source of ATP, right? And glucose is going to be one of 3 monosaccharides. The building blocks of carbohydrates are monosaccharides. So saccharide means sugar. So does amyl. So if you see amylose, okay, is a sugar. Ends in ose is a sugar. If you see dextrose, mannose, maltose, lactose, anything that ends in ose is a sugar. Anything that ends in ace is an enzyme. ASE is an enzyme. OSE is a sugar. That's super important to know. So this is glucose, right? Ends in an OSE, right? So I want you to notice that glucose has made up of carbon and hydrogens, yes? Now, there's a certain number of them that you probably want to know. There are 6 carbons, right? There are 12 hydrogens, and then there are 6 oxygens, okay? So I'm going to count them out for you. You probably don't see them, but they're there. Every time that you see an angle on this is the actual structure of glucose. It makes a ring. Every time you see an angle, that means that there's a carbon. So in organic chemistry, they just take the carbons out and just assume, because in organic chemistry is all about carbon. So whenever you hear the word organic, that refers to anything that's carbon-based. If you're talking about inorganic, that means it doesn't have a carbon base, okay? Which means that it could have carbon in it. It just doesn't have a carbon base. So like, say, carbon dioxide is inorganic, but it does have a carbon. But if it has lots of carbon, hydrogen, and oxygens, most of the time it's carbon-based, and it's basically what we're all built on, carbons. Okay, lots and lots and lots of carbons and hydrogens. Okay, so C6H12O6, that's glucose, right? That's one of our monosaccharides. But we have 2 other monosaccharides, which are slightly different in structure, but they are still going to be C6H12O6. Because when you have this many structures, let's say that you take this OH, and you switch this OH from pointing down to pointing up, that's like actually That's actually the structure. If you didn't know, that's actually the structure of Splenda. So because you actually switch, so this is actually missing an H here, because whenever the H's are, they usually just exclude the H's. These are organic chemists. They shorthand everything. All right. So you have a carbon in the middle. Then this is a carbon bond. This is the carbon hydrogen bond and a carbon oxygen bond, right? Because I told you that carbon makes 4 different bonds, right? So notice how this OH is pointing down and this H is pointing up. If you flip it, it's a different molecule and your body doesn't understand it as glucose. Okay, that's exactly what Splenda is. So in other words, it has to be exactly this way. So there are a bunch of different ways that you could switch these OHs, yes, and these H's, and you're going to come up with a different structure. So glucose is the one that we need, and we need it in this orientation. However, you can have two other monosaccharides. They're called fructose, which is actually, that's a fructose. Fructose, which is a 5-ring, it's a 5-sided ring rather than a 6-sided ring, only because it has the C. You see how the C is kind of sitting up there? So it's 1, 2, 3, 4, 5, 6 C's, and there's an O. Well, it only has 4 C's and it has another one kind of hanging out like that with the C, H2OH kind of hanging off the side. So fructose is slightly different. That one's actually quite different. But galactose is your other monosaccharide. So these 3 are your 3 monosaccharides, okay? So it's very important that you understand that those 3 are monosaccharides. Those monosaccharides are going to be derived from disaccharides, okay? They're going to be derived from disaccharides, because disaccharides disaccharides are going to be made up of 2 of these. Geez, sorry about that. Okay, so you have, for example, galactose, fructose, okay? So this is how this breaks down. I'm going to move this out of the way for just a second. Okay, we'll put it over there. So these are your 3 disaccharides, for example. So let's say that you ate food, right? So you ate food, and your food comes in the forms of proteins, right? Carbohydrates and fats. Those are your 3 macronutrients, right? Carbohydrates are going to be made up of a bunch of monosaccharides. These monosaccharides are going to be composed of either galactose, glucose, or fructose. Okay, those are your 3 monosaccharides that all of your carbohydrates are going to be made out of. All right, so what you're going to have is lactose is a disaccharide, which means lactose is actually going to be 2 monosaccharides put together. Okay, so when you ate when you ate your carbohydrates, they came in long chains of glucoses, galactoses, and fructoses in whatever orientation it is. In digestion, you start to break down. You use enzymes. Remember, they end in ase to start breaking down those sugars, right? We have amylase is going to break down those long carbon hydrogen chains. Is basically what they are: carbon, hydrogen, oxygens. Okay, so you're going to start breaking them down into smaller and smaller groups of sugars. Once you get to the disaccharide, which means it's made up of 2 monosaccharides, you're going to break them down into individual monosaccharides, right? So you have lactose, you have maltose, and you have sucrose, which you probably have heard of, because sucrose is table sugar. So table sugar, regular granulated sugar that you put in your coffee, for example, is going to be made up of a disaccharide made up of a fructose molecule and a glucose molecule, right? So it's a disaccharide. So that's why there's 2 arrows, because when you break it down, you're going to break it down into a glucose, into a fructose. Maltose is actually made up of 2 glucoses. Lactose is made up of a galactose and a glucose. Okay, so that's a good way to remember your disaccharides and how they break down into your monosaccharides. Okay, then they use enzymes, right? So this enzyme here, which a lot of people are lacking, is called lactase, right? Lactase is the enzyme, remember ends in ase, is enzyme that breaks down lactose into galactose and a glucose. So I guess we should probably put it in there, right? So Lactase is the enzyme that's going to break down lactose, a disaccharide, 2 parts, right? The 2 parts are a galactose and a glucose, and it breaks it down into those monosaccharides. In other words, it's kind of like the end of digestion. Now, why is this important? Because when you consume food in these large macromolecules, but digestion has to break them down into the individual monomers in order for you to absorb it. So absorption of nutrients has to occur at the level of the monomer. Okay, so it's not gonna occur as a disaccharide. And you probably know that if you're lactose intolerant. So if you're lactose intolerant, what happens? Which means you have lactose, like let's say that you drink coffee with milk, or you have ice cream. Then what's gonna happen is when it gets to your intestines, because it has to be broken down, right? It's mostly lactose is in milk sugar, right? So it's going to be broken down into the disaccharide. But people who are lactose intolerant don't have lactase. So since they don't have the enzyme to break down lactose into galactose and glucose, the lactose stays as a disaccharide. As a disaccharide, it cannot be absorbed into your intestines. So what happens is it keeps going through your intestines, does not get absorbed into the cells, which means it's going to get absorbed into the blood, just goes through your intestines, goes to your large intestine, and then causes a problem because now you have a very high quantity of lactose, and E. coli is a high lactose fermenter. So it's going to use the lactose. It's probably going to create some. E. coli is also a gas producer. When it breaks down your lactose, it's going to produce gas. So it produces gas. And since you have all of this, do you remember osmosis and diffusion? Okay, so since you have in your intestines, you have all of this lactose, what happens is that the lactose draws the water out of your intestinal cells because they are going down via osmosis. So it basically causes something we call osmotic diarrhea. So it pulls the water out and causes diarrhea. And then, of course, the E. coli that ate some of that lactose is going to create gas. So you get gassy diarrhea. Fun times. Okay? So that's what happens when you can't break down into the individual monomers. So then maltose is broken down into 2 glucoses by maltase. Sucrose is going to be broken down into the 2 glucose and fructose by sucrase, right? So each one of them has their own enzyme. So yes, we have multiple enzymes. So we have something called salivary amylase that is secreted by your salivary glands in your mouth that begin the process of digesting large carbohydrates into smaller carbohydrates. Eventually you have, pancreatic amylase, which continues the process, right? Pancreatic amylase released by your pancreas is going to continue the breakdown of large carbohydrates. Eventually we get down to the disaccharide level, right? When we started with 300, now we're going to get down to, 5 and 6 sugars attached together. You keep breaking them down until there's 2 sugars together, and then you're going to have those enzymes. They're going to then finish off digestion. And then glucose, galactose, and fructose. Now, your blood sugar is made up of all glucose. So in your intestinal cells, we convert the fructose and galactose into glucose. And then now it's going to be basically the rest of your blood is going to be filled with just glucose, because glucose is what we're going to use for energy. So when glucose comes into cell, right, when you eat a lot of food, right, what happens is as the glucose starts to flow through your body, right, through your blood, it's going to then trigger the pancreas to release something else called insulin. And so insulin then is going to bind to a receptor that's found throughout your body, mostly in the muscle cells, because that's where we need the most amount of energy, but throughout your body, everywhere. And they are going to open a channel where glucose comes into the cell, and then glucose is going to be processed into ATP. You're going to make ATP from the breakdown of glucose, and that's what we're going to talk about in chapter 4, right? That's called metabolism, right? So we don't get too ahead of yourself right now. Just know that the breakdown of carbohydrates are going to be into individual sugars called monosaccharides, right? So while this is your one more thing. So we have lots of complex carbohydrates that we consume on a regular basis. The carbs that we can break down have a particular type of bond. It's like a 1,4 carbon bond. But there's some of them that are bonded a little bit different. And those that have a different bond, right? So like the 1 carbon to the 4 carbon is what I was talking about. But if they have a different type of bond, they may not be broken down. So for example, plant cell walls are made up of a carbohydrate called cellulose. We don't have the enzyme to break down cellulose, which is a 1,2 like beta linkage. We don't have the enzymes to break those down. So cellulose never gets broken down in our intestines. We use our acid in our stomach to actually make it so that it's easier for us to move it through our intestines, but it becomes part of our fiber in our diet because it's not going to be absorbed. So I want you to think of digestion as really kind of going through the body, like a drive-through. You're not really in the restaurant. It's not really in your body. It's just going through your body. In order for it to be in your body, it needs to be absorbed into your intestinal cells, and it needs to be in your blood. That's really in your body. All right. So proteins are going to get broken down one way into their individual amino acids, right, by a bunch of different enzymes. And we'll talk about that in the digestive system. So don't worry about that. Then you have a bunch of different enzymes, like I explained here, that are going to break down your carbohydrates. And this one's a little bit more important because we're going to talk about that in chapter 4. But proteins, as I mentioned, some of them are polar, some of them are nonpolar. And carbohydrates, which are made up of a bunch of, carbon-oxygen bonds. So anytime you have oxygen, you have a partial polarity. So sugars aren't super polar, but they have a little bit of polarity. So those are both water soluble. Fats are not water soluble. Okay, so fats are created by the carbon-hydrogen bond, okay? That is the typical carbon-hydrogen bond that you're going to see in organic molecules. Carbon, so for example, this, right? So this is a typical molecule. I mean, this is methane, which is a gas. But what we're going to have in our fats is we're going to have a bunch of carbon. So if this was an H, that would be methane. So that's that gas. But if you see carbon, 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 each one of these has a hydrogen attached to it. Okay. And they could get really, really long. Okay. These are, all of these bonds are non-polar bonds, right? Water is polar. All of these bonds are non-polar bonds. Water uses polarity to break things apart. Okay. So for example, the sodium chloride, right? I'm sorry, I forgot the L. The sodium chloride in salt, once it hits water, what's going to happen is that the water molecules are basically going to take these two things in the crystal and separate them. Because the water molecules are then going to attach themselves as such. Not attach, but they're going to form hydrogen bonds. Okay? Why? Because remember, this one's partial negative and this is positive, so they're surrounded. They form what we call hydration spheres. And that's why when you put salt in water, you separate the sodium and the chloride. And in this one would be the opposite, because here, remember, the H's are partial positive. And so they're going to be like this. The H's are going to be attracted to the chloride, and then the O's are going to be away from it. Okay? So they surround it, and they separate it. That's how water separates things that are polar, because it surrounds the polarity with its partial polar charges. But if it's nonpolar, like fat, no polarity. Water can't do a thing to it. Okay? Water cannot do a thing to it because there's no positive or negative charge that it can surround. Okay? So water and oil, as you probably already know in life, they don't mix. Okay, but this is why they don't mix, because there's no polarity in the carbon-hydrogen bond. They are completely non-polar covalent bonds. So that leaves fat in a unique place, right? So how do we break down fats when we eat fats in our meal? Well, we are going to break down fats because fats do get broken down into their individual monomers, okay? And their monomers are going to be glycerol, which is the 3-carbon backbone, okay, that is going to hold, and this is all H's here. All these separate lines are H's. If it doesn't have anything else, it's an H. So there's an H, 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 right? This is going to be all of these carbons. They look like this, okay? So H's up here. The end of all of those are all H's. There's going to be another set of these here, another set of them here. This is a triglyceride, a glycerol with 3 fatty acids, okay? Looking something like this, okay? A little bit better than my drawing. Okay, so this is your glycerol, and these are your fatty acid chains. Notice that they all have carbons. They're made up of carbon-hydrogen bonds, multiple carbon-hydrogen bonds. Okay, so fats, all of these fatty acid chains are all nonpolar, okay? These fatty acid chains are not going to be able to be processed by water. Water can't break down fats because it can't surround it like it can sodium and chloride and anything else that has a polarity to it. They can break those down by surrounding them with their partial polar charges. There's nothing to attach here. So they're going to be processed completely differently. We have things like, remember, we have, for glucose, we have amyloses, right? In proteins, the proteins are going to be called peptidases, and there's multiple dipeptidases, and there's other ones that have different names, but anything that ends in peptidase is going to be something that breaks down proteins. In your fats, you're going to have lipases, right? So the lipases are going to be able to break down the glycerol and basically detach the 3 fatty acid chains. So basically it leaves the glycerol and the 3 fatty acid chains. But where does that go? Okay, so where does that go is called a micelle or micelle. Let me type that out. M-I-C-E-L-L-E. Okay, a micelle is going to have basically a bubble that is made up of phospholipids. So I don't know if you remember, phospholipids look like that. Okay, so a phospholipid essentially is this. With a phosphate group attached. I mean, this is not perfect, but it has a P, right? A PO4, which is a phosphate group attached. So I want you to notice, remember, PO4, 2-, which is a phosphate, has a negative charge. So what's cool about phospholipids, right? So this is the head. This would be the head. These are the tails. Okay, so this is basically upside down, right? Looks something like that. The head is charged. The tails are not. Okay, so remember, when you make your cell is made of a phospholipid bilayer. The phospholipid bilayer is because you have the polar head on the outside attached to or hiding away the non-polar tails. Non-polar tails, that's why it's a phospholipid bilayer. So it's charged on this side and not in the middle, and charged on this side. So it can interact with water on the outside of the cell, it can interact with water on the inside of the cell, but in between your cell membrane is nonpolar. Remember, I told you that last week. Now, a micelle is a little different because a micelle has only one. So it's not a phospholipid bilayer. It's like a phospholipid layer, right? So a phospholipid layer is going to be where it just has the polar heads on the outside, which can interact with water, and the non-polar on the inside, and all of your fatty stuff that's broken down from larger molecules of fat are all going to be sitting inside. So inside of this micelle, you're going to have the glycerol, which is a breakdown, the fatty acids, which are breakdowns, right? These are monomers. You're going to have also cholesterol, and you're going to have your fat-soluble vitamins, and that's how your micelle is how it's going to be absorbed into your intestines. Now, the micelles stay little because something called bile. So we're going to talk about that in digestion. Bile is pretty cool, but bile keeps micelles very, very little because, you know, if you ever shake vinaigrette, you shake it into a bunch of little bubbles, but then as soon as the bubbles all touch each other, they all start to reform bigger, bigger, bigger bubbles. So the bile prevents them from doing that. That's why it's called an emulsifier, right? But what you really want to know right now is that the breakdown of fats are glycerol and the fatty acids. So sometimes we'll call them free fatty acids. Free fatty acids, so it's abbreviated FFA, right? Sometimes also monoglycerides, which means that it's going to have the 3-carbon backbone, how it was before, but it only has one this fatty acid attached to it. So it's called a monoglycerol. So those are the breakdowns of your three main macronutrients. Now, you also have, right? You also have DNA and RNA. Those are your nucleic acids, right? Your DNA is made up of a sugar, right, backbone called deoxyribose, whereas RNA is made up of a sugar backbone called ribose. So it's a little bit different, okay? And so those two things are going to be the remember that I told you that nucleic acids are basically DNA, and RNA are found in the nucleus of every cell in your body. So if there's a cell that doesn't have a nucleus, it doesn't have DNA. So for example, red blood cells have no nucleus. So that means that red blood cells have no way of fixing any damaged proteins. But other cells that do have DNA can fix damaged proteins because they can make new proteins from DNA. DNA goes to RNA, right? Transcription, and RNA goes into making the sequence of your amino acids, and you can replace your proteins. All right, so that's chapter 2 in a nutshell. You want to know a little bit more about DNA. We're going to talk more about DNA tomorrow. All right, see you tomorrow.